Hello everyone, my name is Salah, and today I'm presenting my abstract, an open source flexible plug-and-play inline CMR image segmentation platform. Our lab is funded by the NIH and we have a research agreement with Simmons Healthineers. In recent years, cardiac magnetic resonance imaging has seen increased clinical use. Yet the time-consuming process of manual contour drawing remains a significant challenge in image analysis. Segmentation usually requires a trained radiologist to manually contour the left and right ventricle, a meticulous process which must be repeated across multiple slices and multiple phases in order to properly assess cardiac function. Machine learning models have been proposed for automated contouring. However, these models typically lack flexibility for manual adjustments and quality control. And while commercial CMR segmentation software has increased accessibility, they are often slower to update algorithms, which has limited flexibility for research, falling short of meeting the evolving demands for CMR imaging. Hence, our goal was to develop and evaluate a platform for a flexible plug-and-play inline CMR segmentation with results readily available on the scanner. The platform needed to be seamlessly embedded within the existing clinical workflow, adaptable to new ML models, and had the option for subsequent adjustment in post-processing software. Our platform was evaluated on a Simmons 3T Vita scanner, which reconstructs images via the ICE pipeline. The ICE pipeline consists of functors which reconstruct the raw data and produce a final image which can be viewed on the scanner console. By using the fire emitter and fire injector functors, we're able to augment the ICE pipeline using a fire server, which allows us to connect to an external system via an SSH tunnel. On the external system, we reconstruct the data and can perform analysis on data acquired and return the images and analysis results, which will be displayed on the scanner console. We implemented in-house reconstruction and segmentation algorithms in separate open-source Python modules, offering flexibility to deploy different ML models. For this study, images were reconstructed using a Resolution Enhancement Generative Adversarial Inline Neural Network, i.e. ReGAIN, which allows us to produce high-resolution cine images from undersampled case-based data. Following reconstruction, the ReGAIN enhanced cine images were segmented using a cardiac segmentation network called Carson. Both image reconstruction and segmentation models are freely available and were chosen to demonstrate the feasibility of the platform. We assessed the feasibility of the automated plug-and-play platform for inline cine segmentation in 308 patients referred for clinical CMR. Cine images were acquired with an accelerated protocol with the following imaging parameters. When the sequence is run on the scanner, the data will be processed in real time, and we will begin to see the contoured images appear in the bottom right corner of the screen. After scan completion, contoured images appear on the scanner console, allowing immediate review and reacquisition if necessary. Left ventricle and diastolic volume, and systolic volume, stroke volume, and ejection fraction were also calculated and displayed on the scanner console. If there is a need for manual adjustments, the images can be exported from the scanner as a set of DICOM files, where the ML-generated contours are stored as a point cloud underneath a custom tag in the image DICOM header, allowing for offline adjustments using post-processing software. We demonstrated the ability to manually adjust these contours by transferring the images from the scanner to Metis, a cardiac image software suite, where a custom add-on allowed us to extract the ML-generated contours from the DICOM header and create a Metis-compatible contour file. The users can then load the generated contour file, which will allow us to review the ML-generated contours within the Metis cardiac imaging software. Users can then make manual adjustments to the contours as needed, allowing for quality control for the automated ML-generated contours. To evaluate the performance of our automated inline assessment, we compared the measurements of our fully automated inline segmentation to the semi-automated offline segmentation. In the fully automated method, the acquired scans were segmented using our ML model and no manual correction was made. In this semi-automated method, the acquired scans were segmented using the ML model available in CBI 42 software and were then manually corrected by a clinical fellow. The two sets of measurements were then compared to assess agreement between the two methods. Linear regression analysis showed strong agreement between inline and offline assessments, with an R-score of 0.96 for the end-diastolic volume and end-systolic volume, 0.92 for the stroke volume, and 0.89 for the ejection fraction. Bland-Altman analysis also showed good agreement between techniques. On average, the model underestimated the end-diastolic volume by 27 milliliters, the end-systolic volume by 22 milliliters, the stroke volume by 6 milliliters, while overestimating the ejection fraction by 6 percentile points. In conclusion, we successfully developed and performed an initial evaluation of an open-source flexible plug-and-play inline CMR image segmentation platform. Due to the modular nature of the pipeline, additional analysis such as strain can also be readily evaluated. Further refinement of segmentation networks may improve agreement with clinical analysis, which can be easily integrated into the proposed platform. The platform allows for rapid deployment of CMR segmentation and analysis, enabling researchers to test the feasibility and performance of new or established deep learning segmentation algorithms in an open source environment. All the code and models used in this demonstration are available on GitHub. Thank you for your attention.